Hello, everyone, and welcome to today's webinar, Design of Ready-to-Use Advanced Crop Genotyping Solutions. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. The rest of your questions will be answered by email at the address you provided during the registration process. But you may also submit any technical questions you have here as well by utilizing that Q&A box, and we will answer you directly. Now, without further delay, I would like to introduce today's speakers. Dr. Glendon Ascough, Research Director, Starke Ayres, a vegetable breeding company based in South Africa. Dr. Fikret Isik, Professor, Fulbright U.S. Scholar, South Africa, 2023, Co-Director of Cooperative Tree Improvement Program, Department of Forestry and Environmental Resources, North Carolina State University. Dr. Vance Whittaker, Professor of Horticulture, Gulf Coast Research and Education Center. Dr. Mitchell Feldmann, Assistant Professor and Director-elect of the University of California, Davis Strawberry Breeding Program. And Dr. Grant Poole, Product Manager, Thermo Fisher Scientific, who will also be assisting with the Q&A today. For complete biographies on our speakers, please click on the Presenter tab at the left of your screen. Gentlemen, welcome. It's so wonderful to have you here today. Grant, you may now begin your presentation.
Welcome to the webinar on the design of ready-to-use advanced crop genotyping solutions. My name is Grant Poole, and I'm a product manager in agrigenomics with Thermo Fisher Scientific. And we're really excited to bring to you today presentations from several of our customers who've been involved with not only the design, but the utilization of these, what we call ready-to-use crop sequencing panels, targeted sequencing panels, utilizing our AgriSeq targeted sequencing platform. Well, this slide really provides the foundation for the rest of this webinar and what the speakers are going to talk about today. So these are the pre-designed, pre-made, made-to-order panels that we've already developed. You can order these from us. You can have these ran at a service provider. We have products that vary in size from a few hundred markers for QA/QC type analyses or trait analyses, all the way up to 5,000 markers for larger scale, high density GWAS, QTL mapping applications. Obviously, we are sequencing a 200 base pair fragment. So that being said, each one of these 5,000 markers or 5,000 amplicons can actually return a series of multiple SNPs that can be used for haplotype analysis or any other sort of downstream analysis. The main KOLs that we've worked with to date include the USDA wheat group, the strawberry group. We've developed some maize products with some KOLs in that field as well as tomato and pepper. Those are probably our top selling panels right now. We've also developed barley, peanut, cotton, canola, and then we are just finishing a soybean panel with the soybean consortia, alfalfa, potato, and bean. We're working with consortia to develop those panels as well. So a wide range of different products that, again, the speakers are going to talk about today. Well, I just want to go into a little more detail of our AgriSeq targeted sequencing workflow. It utilizes the S5 sequencer you see in the top right next to the server, and then the actual workflow you see illustrated in the 5 steps below that. So sample prep can be done with a MagMax Core kit-based extraction or a direct lysis kit at a little lower cost. Traditionally, the panel design was strictly custom, and that's where over the years we have actually began to design more of these what are called pre-designed, ready-to-use, made-to-order panels, which is what this webinar is all about. So we've worked with KOLs and leaders in the industry to develop these panels. And then either the service provider or the lab doing the sequencing will go through a library prep process, basically a sequencing process, which will take less than a day, and then data analysis. It's basically a 3-day workflow that's done in the sequencing workflow. So we really recognize that there was a market demand for a product like this with the pre-designed panels. These were customers that didn't typically have access to SNP marker content, and they really wanted to have pre-made products that were ready to order, and they may or may not have wanted to go through a custom design process. So that's where as a customer, you can try these products, get to know the workflow, get to know AgriSeq as a product, and then see if you want to go into a custom design from there. So our whole catalog or pre-design process was basically made to order, available upon request, and we made these panels with these KOLs in various forms. There's trait panels. I think the wheat panel has 212 traits that are associated with it. We have QA/QC panels for different crop groups as well, for detecting genetic differences and doing QA/QC type metrics. And then high density content, 1,000 to 5,000 markers for GWAS, QTL mapping, and that sort of application. We wanted to make the pricing easy to understand, and we really look to service providers to supporting this business and running this. So we basically offer an end-to-end service. As you're interested in utilizing and running these panels, we can point you to the service provider in your region that can actually get the work done for you at a easy to understand, simple pricing. Here's a slide summarizing the development of the wheat USDA panel. We do not have representatives speaking for this panel today, but I thought I would cover it in this slide. Basically, I can send you information on annotations of these panels, but you can see the average distance coverage, the number of SNPs per chromosome. You see the traits that we have in the trait panels. So in this specific design, there was 5,174 SNPs and indels that covered all 21 haploid chromosomes of wheat. We had 212 trait markers that were correlated to disease, agronomic, and quality traits. And again, this was developed with key opinion leaders at the USDA wheat genotyping labs at Washington State University, Kansas State, NC State, and NDSU. We also have a updated soybean panel coming out that was intentionally made to be 6K. This is a new and improved soybean panel that we made in conjunction with the Soybean Consortia group. They recently finished a Williams 82 near gapless genome. As you know, Williams 82 is a very popular reference genome for soybean. This is a new and improved panel. We also have about 135 trait associated SNPs with this panel. And I think that one thing that we had in mind with developing this panel was with the discontinuation of other genotyping products out there, I think the longevity of this product will be around for a long time. And again, it is updated to the near gapless genome, as well as updated annotations for the trait associated markers. So we're excited today to have several speakers who have not only developed these AgriSeq targeted sequencing products, but from what you'll hear, they are still in the development phases of these, of refining their breeding programs, moving from high density 5K panels down to 1.5K genomic selection and genomic prediction type breeding applications. So you're going to hear from Dr. Glendon Ascough of Starke Ayres in South Africa, Dr. Fikret Isik, a longtime pine breeder at NC State, and then Dr. Vance Whittaker and Dr. Mitchell Feldmann from the University of Florida and UC Davis to talk about their development of the strawberry 5K and 1.5K panel. And with that, I'm going to hand it over to Glendon to talk about his experience using the maize and tomato pre-designed AgriSeq panels at Starke Ayres in South Africa. Glendon?
Thanks, Grant. Delighted to be able to share a bit of our experience using the tomato and maize AgriSeq panels. A little bit about our company for those who might not be familiar with Starke Ayres. We're a family-owned vegetable seed company, been operating since 1877, but doing our own breeding and R&D activities since the mid-1980s. We work on 9 different vegetable species, 7 primary R&D locations within South Africa, pathology team of 5 and a biotech team of 8. These are the crops that we're actively breeding and trialing around the beautiful country of South Africa, and the diverse environment from hot, humid in the northeast right the way down to Mediterranean in the south allows us to breed for all kinds of climatic conditions. Our biotech lab activities: we do quality control of hybrid and parent seed productions running between 8 and 15 SNP markers per sample, marker-assisted selection for our breeding team, anywhere from 1 to 30 trait-linked markers depending on the crop, marker-assisted backcrossing with the trait-linked markers and a fingerprint of polymorphic markers for the background. And then we do a single plant genotyping where when a breeding line has finished its inbreeding process, we want to understand the full breadth of its genetic fingerprint, and so we will run anywhere from 100 to 150 SNP markers, kind of everything we have in the lab on those individuals to help us understand the genetics in the fingerprint. So we're a mid-throughput lab running anywhere from 4.5 to 5.5 million data points and processing around 150 to 200,000 samples. We do some whole genome sequencing, which is outsourced to third-party providers, and we make use of the sequence data ourselves in-house with our bioinformatics team. But what we really lacked is a high-density fingerprint, anywhere from 500 to 5,000 markers. And this is the main reason that we got on board with Thermo Fisher and ran the AgriSeq tomato and maize panels. The tomato panel is a 5,000 SNP panel and 21 trait markers. The maize panel is 3,600 SNPs, 160 markers for QA/QC, and 259 for trait link markers. So firstly, really good quality call rate. We were very happy with the quality of the data we got back from Thermo Fisher. 4,900 of the 5,000 tomato markers had an 80+% call rate. As you can see, nearly 4,300 were above 90%. How have we made use of the data in our breeding programs in our R&D? An example from the maize, we use a genome visualization tool called Persephone, and we're able to import the VCF files directly from the Thermo Fisher data into Persephone, and it kind of displays like this. And so this is chromosome 1 of the NAM5 maize reference genome. The little green lines along the top track are the different gene models. The middle track are the trait-linked markers and their positions on the genome. And the last track are 5 different sweet corn lines that were genotyped on this panel of markers. If we zoom into one of those markers, we're able to see the gene model on the top. We're able to see the exons and the introns and the precise location of these particular SNPs and the surrounding and flanking region that is also sequenced, and we can see that there is some variation in our sequenced lines. When you zoom in a bit further, we're able to see that some of these SNPs are in the intron region, while 2 of them are in the exon, the coding region. This SNP has no variation within this small panel of lines. Blue is like the reference genome, red indicates a SNP that is an alternative allele. And beautifully, what we're able to do is have a look at which SNPs are having an effect on the protein. And so this particular SNP in these 2 lines has an amino acid substitution. The SNP has resulted in a functional change in the protein. And this has helped us understand on a very practical allelic level the differences in our material and how they relate to the particular trait or the particular gene that we're focusing in on that time. Example from tomato of how we've used this data. When we run our full panel of markers in our own lab, which for tomato is 129 SNP markers, this is an unrooted tree of 380 breeding lines from our program. Typically, we can look at them like that. When we run the exact same panel of genotypes on the 4,000 markers after eliminating a few based on call rate, etc., we have a much clearer picture of our germplasm and which lines are more closely related and which lines are further or more distantly related to others. So it's really clarified for us our germplasm and its relatedness. We're also continuing to use the data. It's not a once-off, and so we're actively looking at genomic prediction aspects and models, QTL mapping, and we're looking to design some custom panels for other species. Genome-wide association studies are just starting for sweet corn and tomato with this data set, and we'll be excited to add our own proprietary trait markers to subsequent panels that'll be run for us. So I hope that gives you a little picture of how we're using it. And now we'll hand it over to Dr. Fikret Isik, a pine breeder and global consortia leader at North Carolina State.
Thank you, Glendon. We conducted a pilot study to evaluate a targeted GBS panel for loblolly pine in collaboration with Thermo Fisher Scientific. The objective of the project was to develop a cost-efficient genotyping platform for quality control in pine breeding program in the southern United States. So for the project, we selected about 1,000 high-quality markers from Axiom Array with all for the pine, and we amplified those high-quality markers, and quality control check was performed against the loblolly pine reference genome. The markers that passed the quality control were then submitted for the primary design phase, and during that phase, oligo candidates were generated and filtered, and the most optimal oligo pairs for each marker were selected for the final panel. So in total, we designed about 1,000 markers and yielding a 98% design rate. That was very good result. So this table shows the quality metrics for the panel. You see the panel metrics on the left side, the first column, and then statistics for the population we generated. The sample call rate was almost 86%, and uniformity 89%, sample on target 93%, and average coverage was over 200x, which was really more than we needed, but it doesn't hurt to have more data. So all quality metrics indicate a well-performing GBS panel for pine with the population we provided. So we validate the performance of that panel using a pine population, about 192 samples. We generated genetic relationships between samples using AgriSeq panel, which is on the right, and also pine Axiom Array, which is on the left. Those are the heat maps of genetic relationships between samples. I'm talking about the upper diagonal of the matrices, and as you can see, they are very similar in terms of calculating genetic relationships between samples. So The genetic relationships calculated from AgriSeq panel also performed very well in terms of average inbreeding in the population. And you see the average inbreeding from different platforms, from pedigree, from SNP array, and then AgriSeq at the bottom. AgriSeq average was very similar to the Axiom array, which is in the middle. We also looked at the average genetic relationships between samples. Those are coefficients of relationships. Again, AgriSeq panel, which is about 1,000 markers, produced similar average as Axiom array, which has like 50,000 markers. So we were very pleased to see similar statistics in the population. So genetic relationships calculated from SNP markers, either SNP arrays or AgriSeq, they have a lot of downstream applications in plant breeding. And this plot shows clustering of 8 fossil families when we used SNP array on the left, and then when we use the AgriSeq on the right. Again, you can see the cluster of the fossil families. The AgriSeq performs similar to SNP array. Maybe the next slide, explains the power of AgriSeq in terms of quality control. Again, in this plot, you see 8 fossil families with different colors, and then 2 individuals are not grouped with families based on the pedigree. So those are obvious pedigree errors in the records. So AgriSeq panel was able to capture that problem in the pedigree, and we used that information actually to correct our pedigrees. So we plan to use that panel for other applications, including QTL mapping and GWAS, but also use the imputation methods to increase the number of markers for genomic selection. Since we have a high density linkage map, we can do that using AgriSeq panel. So overall, we were very happy with the product, and Thermo Fisher was very cooperative, very helpful in this pilot project. And now I will hand it over to Dr. Mitchell Feldmann, assistant professor at University of California, Davis.
Thank you for your presentation, Dr. Isik, and thank you to Thermo Fisher Scientific for sponsoring this webinar and for inviting me to speak. And also thanks to LabRoots for hosting. My name is Mitchell Feldmann, and I'm an assistant professor at UC Davis, and alongside Stephen Knapp, the director of the UC Davis Strawberry Breeding Program. The work I am presenting today was only made possible by the shared collaborative effort between our program at UC Davis and the University of Florida Strawberry Breeding Program led by Vance Whittaker, who will be sharing details on the second stages of our work in the next talk. I also want to give a special shout out to Josh Sleeper, who has been really taking the lead on the second phase of this project over the last year, which has been wonderful. But today I'm going to be telling you really about the design of medium density genotyping platforms for octoploid strawberry, at least from our perspective. None of the work that I'm sharing today would have been possible without the support from our collaborators, sponsors, and funding agencies. Over the last 9 years, UC Davis has been at the forefront of genomic resource and tool development, including multiple genotyping arrays, several medium density genotyping options, ultra-low genotyping like CASP and TACMAN markers, Illumina and HiFi resequencing, and of course, reference genome development and annotation. All of the things that we've learned from these different exercises over the last 9 years have really impacted how we approach thinking about medium density genotyping and marker selection today. First, I'm going to tell you a little bit about the global strawberry industry, focusing on the US strawberry industry. Around the world, strawberries are consumed in almost every country and play varying roles that sort of lean between cultural, traditional, health-based, and enjoyment as bringing the value. They're not a calorie crop, so you're not eating strawberries to survive, you're eating them to thrive. They're nutrient dense and rich in antioxidants and vitamin C. Around the world, 9.2 million metric tons are produced. About 30% come from China, 20% from the United States, and in the United States is a $3 billion market cap. In the United States, about 90% of the production comes from California and 10% from Florida. Strawberries are produced in many other states as well, but those are the primary two strawberry producing states. And that difference is really driven by different environmental conditions that provide longer or shorter growing seasons that are capable of producing high quality berries. Our industries consist of nurseries that produce clonal plants and ship them to coastal growers after harvest. These are then planted either in the fall or early summer, depending on the market segment. And fruit are typically harvested from January to November or December. Again, this is spanning the entire year, and that includes the different market segments. California's Central Coast growers are primarily focused on long shelf life and long distance shipping and of red fruited varieties. So most of the year, if you see strawberries that are coming anywhere in the United States from Michigan, Minnesota, Maine, and also the South, a lot of the strawberries are going to be coming from California. And so we want to be able to meet all of those consumer demands across the United States with uniform, consistent products. And so to meet the evolving demands of growers, packer shippers, retailers, and consumers, we've been collaborating with the University of Florida, working on developing and deploying marker-assisted selection and genomic prediction across different stages of our different breeding programs. However, the offerings today have been cost prohibitive. Ideally, we want to be able to produce genome-wide marker data on 10 to 20,000 individuals per year, but it's really not possible to do that as an academic, as public programs, when the genotyping options cost $60 or even $30 per sample. So we've partnered with Thermo Fisher to develop a catalog panel using their AgriSeq technology, and I'll talk a little bit about that design process now. Again, we developed this Axiom array back in 2018, and we've genotyped about 18,000 accessions with this platform since then. So we have a really large backlog of data that can tell us what markers are better than others, which ones are segregating amongst the parents that have been historically used, and to filter through so we can sort of deprioritize or prioritize markers before we get into sort of the selecting for a reduced set. So I wanted to select 5,000 SNPs from these Axiom arrays that would capture the genomic information that GBLUT models would be exposed to in the genomic relationship matrix. So I ran a simulation where basically 5,000 markers out of 50,000 were selected at random, and then calculated the genomic relationship matrix between those 5,000 markers, or captured by those 5,000 markers, and then compared that using R-squared and RMSE to the full 50K genomic relationship matrix. And so what you see on the right is the example of the best one after some fidgeting. So after identifying the set with the best R-squared and the lowest RMSE, I went back and tried to basically declutter, so regions that were about a megabase that had many markers in it. I moved some around because the LD landscape in strawberry is not so short that you need that many markers in a small region, and tried to move them out to sort of fill some gaps. So I tried to declutter and gap fill. Following this, I again checked to make sure that we didn't diminish the value of the platform. And so again, that's what you're seeing here on the right. In the top inset panel is sort of the RMSE from 2,500 of the 20 million iterations. I didn't want to plot 20 million because the x-axis got really ugly in base R. But you can sort of see that there is a nice curve of better and worse performance. We then wanted to convince ourselves that these markers and would be functionally equivalent GRMs would achieve the prediction accuracies that we've hoped. And by achieve the prediction accuracies that we hoped, I really mean achieve the prediction accuracies that we've seen. in existing data. So I tested 3 different conditions with 500, 5,000, and 50,000 SNPs. The 5,000 set is the set we just talked about. I did the same thing, but used 500 instead of 5,000, using simple cross-validation for 7 different traits and 2 different population conditions. On average, the 5K SNP platform performed equally to within 0.01 or 0.04% accuracy relative to the 50K version. So again, if you make the assumption that you can perfectly type all of these SNPs using a different technology, you'll be able to achieve effectively the same genomic prediction accuracy using them. So again, it puts sort of the burden on the technology because we're starting with high quality informative SNPs for the purpose that we've intended. So we're really excited to see this platform come to life, and I'm happy to pass the proverbial microphone to Dr. Vance Whittaker from the University of Florida to tell you more about the next steps that we've taken.
Thank you very much, Mitchell. Our group here at the University of Florida in strawberry breeding have been beneficiaries of all the great work done at UC by Steve and Mitchell and their team. We really appreciate the Axiom Array and the 5K AgriSeq panel that we've been working with so far. We really appreciate all their work. And what I'm going to present here today is the work of Josh Sleeper, one of the members of our team here at UF, who has been working with UF and UC SNP data from the AgriSeq and from Axiom to try to optimize a final panel for strawberry that optimizes marker numbers and specific markers for breeding applications. So next slide. When attempting to optimize marker density in strawberry, it's tricky because strawberry is an allooctoploid with 28 chromosomes in the haploid. It's highly heterozygous, and so there's a large recombination space. And so we couldn't make a lot of assumptions. We really had to go about this empirically. So what we did here is we took Axiom SNPs, which is always our starting place since these AgriSeq SNPs are a subset of the Axiom. And what we did is we looked at a number of markers on the x-axis here, and then prediction accuracy on the y-axis for 3 different traits: average fruit weight, culls, which is unmarketable fruit, and marketable yield in UF strawberry breeding program. And this is the most difficult scenario where we're predicting using genomic selection new individuals and new environments. And it's also in cases of predicting within biparental crosses, predicting seedling performance within biparental crosses, which is also the hardest breeding scenario for us to get high prediction accuracy. So this is the toughest case all the way around and gives us a great test of these marker densities. And what you can see here is that prediction accuracy is starting to level off around 1,500 markers. And you can also see, based on the color coding there, that imputation up to the larger number of Axiom SNPs on the SNP chip is always helpful, regardless of the marker number or trait. So what we're seeing here is that we need to aim for around 1,500 SNPs in order to have the best number of markers at probably the lowest cost that we can in the final AgriSeq panel. And so that's what we're going for. And that imputation is something that we're going to be regularly doing in order to maximize and optimize what we're doing. What I'm showing here is Josh's work with SNP calls from UC and UF materials that have calls on both the current AgriSeq panel and Axiom. And what we found out right away is that we already had about 1,900 SNPs with greater than 90% concordance between the two platforms. So that was a great starting point, especially considering that these concordances did improve with additional runs and should continue to improve with additional runs in AgriSeq as we build up that database. So we started with around 1,500, but then added further SNPs in order to fill gaps. We wanted to make sure that we had a marker at least every 2 centimorgans. We also, each program added locus-specific tests, about 50 of those, to ultimately get to program-specific panels of about 1,650 SNP loci. And we really do appreciate the ability to customize our breeding program's set of AgriSeq SNPs with those locus-specific tests. Grant and his group have been very good to work with in that regard, and we just appreciate the flexibility of the technology to allow for that. So the verification is ongoing. We do need to do larger runs with our current DNA extraction methodologies and current sequencing providers in order to look at our scale, how we're doing at scale, turnaround time, and so forth. So the verification is ongoing, but we're very optimistic that we're going to get to where we need to get to. Because really our ultimate goal in our breeding program is to really apply this at scale, because we're applying genomic selection at all levels of the breeding program, parent selection, cross-combination selection, seedling selection. And so we really want to apply this at scale and use the array now for smaller numbers of individuals, important breeding parents, and so forth, to really help with our imputation. But we really look forward to continuing to work on this and hopefully using it moving forward. Thank you so much for having me. And at this point, I'll throw it back to Grant for Q&A time.
Thank you, gentlemen, for your insightful presentations, and I'd like to welcome you all back onto camera for our live Q&A. So we do have some interesting questions from our participants that have already come in, but to our audience, if you haven't already done so, we invite you to submit those questions now. Simply type them into the Ask a Question box and click Submit, and we will answer as many of your questions as we have time for. So now I'm going to hand it over to Grant for our Q&A section.
Yeah, thank you, Christy. So yeah, we're delighted to have all of the speakers on this webinar. Thank you for joining today. I just want to take a few minutes to acknowledge a few people that I see in the audience. So you heard me made reference to the soybean panel. I see Henry Nguyen, his group actually contributed significantly to that soybean panel development. That panel is being validated right now, but I want to acknowledge Henry and his group in that work. Also want to acknowledge our internal team that has helped all of these customers, including Srinivas Shataram, who I work closely with as a senior product manager for AgriSeq. He has been involved in leading this business since 2018, so he's been very influential and instrumental in all of these development projects. And then our bioinformatics team, we have about 4 bioinformatics scientists that will help with design. They'll help with custom design, and again have helped all of these customers design these projects that take, as you know, when you get into genotyping, the timeline can take quite a few months. So want to acknowledge all of those people. And then also North American Genomics has been a service provider, as you've seen in the chat, that a lot of these customers have used and appreciated over time. So just wanted to make those acknowledgments. And from there, I think we'll go into our first set of questions. So we do have a question that I think cuts right to the chase here. And I don't know if our speakers have seen this, but we have a question from Felipe regarding reproducibility. Have you tested reproducibility? For example, when the same individuals genotyped multiple times, on average across all SNPs, what percentage of the time do they output the same genotype? So I'm going to actually turn this over now to Mitchell, Vance, Glendon, Fikret. If any one of you want to field that question, I know that some of you have actually made mention to this in your presentations. Feel free to respond.
I'll take a stab at it. Sorry, everybody. The concordance between individuals that we've tested at UC Davis has been pretty high, and especially if you have SIB information and parent information, you do a really good job of assigning individuals to the right parents, which is promising. Vance has also looked at larger numbers of replicated individuals than we have, which spurred us to start doing that a little bit more frequently. And so Vance might have some more specific details from his experience, but for us, it's been pretty good.
Yeah, Felipe, I would need to dig into the data, but I'm confident in saying that most of our duplicates of the same genotype return call identities above 90%, most of them above 95%. So it's very similar to the kind of concordance data we showed between AgriSeq and Axiom.
Yeah, very good. And Fikret, have you seen the same sort of concordance values? I think you made mention to it in your presentation as well.
So we tested a small sample to see the reproducibility. We are still actually working on the data, but it looks like promising so far. What I know from the preliminary data analysis.
Very good. Yeah, so I like to lead on our speakers as customers to answer those questions, Felipe. But from Thermo's perspective, I can tell you that every customer that we work with has this question and this issue, and our bioinformatics team can actively step you through that process. If you send us data, we can promptly get back to you and with those sorts of analyses. So we've helped Vance, we've helped Mitchell, we've helped Fikret and his team figure out those kind of questions. So that's where I think when you come to Thermo Fisher and you run these products, you will find that you have that support to answer these sorts of questions and look at the functionality of the product, how it's performing. We're always refining it and trying to work with you to make it better. We've got several other questions coming in here, and I haven't read these yet. I'm just going to read them out. So what have the speakers found as a single greatest hurdle in running AgriSeq? Anything from selecting SNPs up front for the panel to data analysis at the end. Does anyone want to field that question?
Maybe I can, take a step at the question. For Pine, the major hurdle was to design primers for those SNPs we selected from SNP array. Unfortunately, Pine reference genome is very fragmented, and it makes it difficult to design primers and also oligos for those markers. So, but overall, the quality we got from the design in the panel was very good, and we were happy with the final results. And Thermo Fisher bioinformatics team is very skillful, very helpful, and they work with you, and they were great in terms of helping us to come up with the panel.
Yeah, thanks, Fikret, and I'll just comment there as well. So with the pre-designed panels, this is one of the hurdles that we tried to cross for you as a customer. So working with these KOLs and working with our bioinformatics team, we actually tried to refine these panels, and as Glendon and other customers have ran them, we continue to refine them for the customers. But these are pretty much ready to order, ready to go pre-designed panels. But we have a whole custom side of the business that will help you design and go through the design process that all these customers have gone through as well. Okay, I think I'll move to the next question. With strawberry, if the 1.5K panel is applicable to genomic prediction, that is a question. Is the 1.5K panel applicable to genomic prediction? Will ultra throughput trait markers still be useful to screen seedlings at a large scale? This is from Navneet at Fight for Genotyping. Anybody want to answer that, Vance or Mitchell?
I can take the first stab, and then Mitchell can add if he likes. I think the bottom line is yes, because while the AgriSeq panel is certainly more cost effective than the large Axiom array, running HRM or CASP assays for locus-specific tests is on the order of cents per sample. So we're probably never going to be able to run an AgriSeq on every single individual in our program, and so we're going to have to pick and choose a little bit and still use those locus-specific tests. particularly in certain seedling applications. So I think they all have a place, right? So Axiom for genotyping our most important parents, AgriSeq for genotyping the most important families and individuals for genomic prediction, and then locus-specific tests via HRM or CASP, in-house, or in order to screen even larger numbers of samples for certain populations.
Yeah, that's a very good answer. So I just want to add to that. We actively take CASP or TACMAN assays that are developed for specific targets that are associated with traits, and we routinely include these in an AgriSeq panel. With AgriSeq, the workflow is much more streamlined. You can run so many more samples over those panels. So for Vance and for Mitchell, they have their own proprietary trait-based panels that are brought into AgriSeq. The advantage that you get over a qPCR assay with AgriSeq is that 200 base pair fragment. And so not only do you get a genotype call around the SNP, but you get everything, any other type of variant information in that 200 base pair fragment. So from there, I'll go into question number 11 here. We have from Dongyan Zhao that asks a specific question around what can be extracted from that 200 base pair fragment. And so I think that question segues well into what we were just speaking about. So what our design and program and AgriSum toolkit analysis tool does on that third day of analysis is it takes each 200 base pair fragment and aligns it to the reference genome. And from that, we get your indel, your large variant analyses, and returns the variants that we see there. You also get novel variants. So each 200 base pair fragment, when our design team, our bioinformatics team designed that, it's usually done with one SNP in mind, right in the middle of the 200 base pair fragment. But what we often see is that our users will find novel variants. You can find as many as 5 or 6 novel SNPs or large indels in those as well that we can pick up and detect. So hopefully that answers that question. We're getting a lot of questions. This is great. I love the interaction here. And again, as Thermo Fisher, this is great because we interact with so many global customers. It's good to interact on this platform. So number 12, is the output in BCF format? What software tools are available to export specific SNP genotypes in an Excel-like format? That's a great question from Michael Havey. So the answer is yes. So we can return in various formats, including a BCF. We also return in an Excel-based reference variant call. So you'll get the reference allele, a slash, and the variant allele in what we call a matrix coordinate type Excel file. And then we also return it in a top-bottom format, similar to what maybe Illumina does with the AABB genotype calls. So there's various different calls. We can also give you the bed files. We can give you the actual 200 base pair fragment sequences. So we can return a lot of different types of data, and North America Genomics and other service providers can help that as well. Number 13, is anyone using pooled samples to get an overall frequency of variants? Or are groups exclusively for genotyping individual plant samples? This is from Camille at Lando Lakes. So I would say that, and I understand for certain crops like alfalfa, sometimes there's open pollination and different types of different reasons that you would want to pool the variants into a single well. The answer is yes, we can do that, and we're actually working on an alfalfa project where we're doing that right now. So I think we can take that question offline and get into some more of the details there later. From Patrick Woods at Plant Sciences. Hello, everyone. Thank you for the great presentations. My question is directly for both Vance and Mitchell. So get ready, Vance and Mitchell. Do you believe the Strawberry 5K panel is sufficient for GWAS QTL mapping projects, or would you advise to use a denser 35K or 50K SNP array for marker discovery projects in Strawberry? Vance, Mitchell?
I can take this one first and then let Vance answer second. So we've done some genetic map creation with the 5K panel, and you are able to produce genetic maps. Right, strawberry has 28 base chromosomes, and so you don't have a huge density of markers throughout the chromosomes, and so depending on the patterns of inbreeding, they may be more or less dense and sparse in regions of interest. So from my perspective, it's not something that I'm thinking about using for QTL identification and discovery projects, but is really a tool for routine genomic evaluation. I think if you have a really large effect locus, you will have markers on this 5K panel that are linked to that. And in fact, you'll have markers on the 1.5K panel that are linked to that because of the way that Josh went through and made sure that we have a marker effectively every 1 to 2 centimorgans of the strawberry genetic map. So it's possible, you just won't have the same type of resolution.
Yeah, and I agree with Mitchell that probably this panel will work well for you to detect, for example, via GWAS, any locus that has an effect large enough to be breeding relevant. You will probably find it with this 5K panel, just based on the recombination landscape in strawberry. I don't think you necessarily need, especially given that the imputation accuracy to the Axiom array is going to be 96, 97%, based on our, so you can genotype with a lower density impute and then run your GWAS. That's another approach to consider.
Yeah, no, those are both very good responses there. I just want to add to that and say that when we developed the maize panel, I'm thinking of specifically, that was a 3K panel. We actually used minor allele frequency, pick values where we could to find those best performing markers that are going to give us the highest level of polymorphisms. And I can tell you anecdotally, Glendon actually ran those panels and reported on that in his presentation. But we found out of a 3K panel and global customers running samples over that panel, because we use minor allele frequency and the frequency that's in that panel is actually greater than 30% when we designed it. Because of that, we're seeing up to 1,800 to even 2,000 markers that are polymorphic in populations that have been returned. So it's all in really marker selection, how you go after those markers, and Vance and Mitchell and Josh have worked together to do a good job of doing that, going from the array level down to the 5K panel, and then obviously on down to the 1.5K panel. We've got a question here from Jerry Fiedelson, Hudson River Biotech, and we know Jerry, so thanks for joining us today, Jerry. Thank you for the difficult question. I actually think this is a very good question to answer. So he asks, a somewhat surprising outcome of selecting a subset of catalog AgriSeq panel markers in a much smaller trait panel on another population is the high percentage of monomorphic markers. Most of these can be rescued by Thermo Fisher identification of novel markers in each amplicon, an excellent advantage of this technology. So actually more maybe of a comment rather than a question, but yes, this is a case of where we worked with Jerry to produce a smaller set of markers, but we were able to, where we saw certain amplicons that were returning monomorphic calls, we went into that 200 base pair fragment of those amplicons that were already linked to the trait of interest. I think in this case it was disease resistance, and we were able to find variants in those amplicons in the samples that could potentially be used as a marker. And I can tell you, working with other customers where they've had gene regions of 2 kb to 5 kb that were linked to a trait of interest, we've been able to go into NCBI, take the sequence from that region, and our bioinformatics team is very experienced in tiling and making tiling designs across those. Basically, customers can submit samples, and then we can look at the variants that we find in those in those amplicons. We can take 30 amplicons, for example, across a 2 or 3 kb region that are 200 base pair fragments each, and then we look for variants, and we basically line those variants up with the trait of interest. So we can do that kind of work and help do some of that work as well. We just, in that sort of situation, you need a good phenotype on the individuals that you're actually genotyping. So with that, I think, are there any other questions? I don't see any other questions coming in. Is there anything else from the rest of the speaker panel? Thanks again, all of you, for joining today. Anything else from the speaker panel that they want to comment on or say?
I just want to comment on using AgriSeq panel for genomic selection. Genomic selection simply captures genetic relationships between samples better than pedigree records. And most of markers are neutral, and they trace those haplotypes, segregating in the families. We are testing that concept in pine breeding, but we believe that if we impute the genotypes from AgriSeq panel, which is about 1,000 markers panel we have, maybe up to 5,000 using the linkage map, we believe it's going to work even for pine, which has a very large genome.
Excellent. Thank you for that, Fikret. No, that's a great comment. So we do have one more question that came in regarding obtaining SNP information from the flanking region of the target SNP. So somebody must have heard me explain how we're sequencing that 200 base pair fragment. What will be the genomic window size for getting the flanking SNPs? Will it be 200 base pairs or more than that? Very good question from Jinli Kim at University of Florida. So basically, as I mentioned, yes, the short answer is yes. Each fragment is going to be 200 base pairs, and we can extract all of the information. Those variants will be automatically called in our AgriSum toolkit. So both the hotspot that's right in the middle of the amplicon will be called, as well as the novel variants, and we'll return those in Excel files to you. What we can do is take those novel variants, or even the indels that we find that are in addition that we weren't expecting to see, and our bioinformatics team can basically redesign the hotspot and region call files. So as you run future samples, let's just say you run them at a service provider like North America Genomics, we can give them those files and they can automatically call those. So for instance, with a potato panel that we've worked with, there was 3.4 thousand amplicons. We were able to call upwards of 11 to 15,000 variants in those amplicons, and we can have our design team redefine those call files. So when you get an Excel matrix file back, you get all of those genotype calls across, several calls within each amplicon, if that answers your question. The other aspect is the tiling design that I mentioned. So let's just say you have a 2 or a 5 kb region that you want to characterize. We will design 40, 50 amplicons across that region to basically tile that entire region. And then we can look at the variants from all of the amplicons across those regions. So I think with that, I'll wrap it up. I think that what I want to put in perspective here, though, in all of the speakers' presentations, is that whether you're looking at Axiom or AgriSeq, what we do well here at Thermo Fisher is characterize targets. So if you're coming from whole genome sequencing, or you're coming from a discovery type project, or even from microarrays, where you're looking at thousands and thousands of variants, our real objective is to get you down to a few thousand variants that you can screen on a routine basis at a lower cost. That's what our bioinformatics team will do. That's what these customers have done. Thanks again for presenting today to all of you, and I will hand it back over to Christy once I think we're finished.
Wow. What a great webinar. A big thank you to Dr. Glendon Ascough, Dr. Fikret Isik, Dr. Vance Whittaker, Dr. Mitchell Feldmann, and Dr. Grant Poole for your presentations and this informative live question and answer session. Now, before we go, I do want to thank our audience for joining us today and for their interaction and their questions. Any questions that come in during our on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Today's webinar will be available for unlimited on-demand viewing shortly. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your friends and colleagues who may have missed today's live event. Now, I'd like to bring your attention to a survey at the left of your screen. We'd be grateful if you spent a couple minutes answering the questions. It will help us learn if we've covered your expectations and to improve your experience in upcoming webinars. As always, it's such a privilege to collaborate with our partners here at Thermo Fisher Scientific. Everyone, thanks for joining us. Until next time, take care. Bye-bye.
Thank you.